Intracellular bacteria secrete effector proteins into the host that can manipulate biological pathways to promote pathogen survival. Revealing host targets of effectors is essential for understanding intracellular pathogens like Chlamydia trachomatis. Yeast toxicity and suppressor screens can provide key insight regarding the natural biological target of bacterial effector proteins and can be particularly useful when a binding partner is unknown. We demonstrate the yeast toxicity and suppressor assays here for screening Chlamydia trachomatis effector proteins, but this technique has also been used for characterizing Legionella pneumophila and Coxiella burnetii effectors.
Begin by inoculating 5 milliliters of single dropout broth with a single colony of yeast transformed with the effector protein containing plasmid. Use yeast transformed with the vector alone as a negative control and incubate the inoculum overnight at 30 degrees Celsius while shaking. On the next day, add 180 microliters of sterile water to the A2 through A6 wells of a 96 well plate. Vortex the overnight culture and add 180 microliters of yeast to well A1. Then, serially dilute the yeast in the wells with water. Use a multi-channel pipette to spot 5 microliters of each dilution onto single dropout glucose and single dropout galactose agar plates and incubate the plates at 30 degrees Celsius for 48 hours. After the incubation, visually assess toxicity by comparing the growth of yeast expressing the effector protein grown on the galactose-containing media to the growth of yeast expressing the vector alone. Inoculate 100 milliliters of the single drop